Hello, my name is Evelyn Walton and I'm here to show you this new invention, a DNA extractor. This was created to help achieve Sustainable Development Goal number 16, which addresses peace, justice, and strong institutions. This invention will help to work towards this goal because when investigating a DNA fingerprinting case to solve a crime, DNA needs to be extracted from cells in order for the process to work. The initial extraction of the DNA is the most tedious and time-consuming part of the entire process. By using this invention to extract DNA instead of doing it manually, the crimes will be solved faster. The steps to extract DNA are as follows. 1. Retrieve the DNA from the cell. Then, you get rid of all the proteins and cellular debris that are on the cell and DNA. Then you obtain the precipitate of the DNA strands, and then clean the DNA strands and check that the DNA is good enough for use. In this invention, all of these elements are combined. The cell is put into the DNA extractor, then salt and detergent is poured into the top to deconstruct the nucleus and cell membrane so that the DNA is released. Then, the cell goes through the filter here at the top, which is one of the ways to get a pure DNA sample. This can also be done by adding a protein enzyme. After the DNA goes through the filter, ice-cold ethanol or isopropanol is added to the sample to get the precipitate of DNA strands because DNA is not soluble in alcohol. The DNA then gets purified further and put into a mildly alkaline buffer. After this, the DNA sample needs to be checked to see if the density, concentration, and purity of the DNA is good enough to go through the electrophoresis process, which is what is used for DNA fingerprinting. The ready-to-use DNA sample comes out on the other side of the extractor and then can be used to help solve crimes.